So welcome people at home, welcome people here. And yeah, tonight's presentation will be the soil food web under the lens with Laurie Dixon. But before that, we've got some things to talk about. So thank you all very much for being here. I'm Mark Stetzer. This is the Williamson County chapter of the Native Plant Society of Texas. Very good. For those attending in person, tonight's plants, as you saw back there, we've got Demi Anita, nice evergreen, doesn't like water once established, and Texas tuberose. And also we had 3 Esperanza come back. I think we're down to 2. So that's good. Help yourself back there. Great plants to have in the garden. Upcoming programs, November, Pocket Prairies, and then we have our annual holiday party at Georgetown Public Library, and then Dark Skies and Native Plants in January, and then some macro photography of native insects in February. So good programs coming up. Tune in from home or see you at the library. And I just want to say thank you all for being here and paying attention to the blog posts, as well as the website, as well as the email. I'd really like to thank our secretary for getting that taken care of, and then Pat Donica for getting that information out. It was super fantastic to have that happen at the last moment so quickly and so smoothly. Upcoming field trips. Berry Springs, October 26th, 1:30 to 4 p.m. Godwin Preserve Plant Survey, November 16th. Gary Park, November 30th. And then the annual New Year's Walk, to be determined, actually will be on January 1st. So that'll be fantastic. Just to recap, September 12th field trip, native grasses with Ricky Lennox at River Ranch County Park. I don't know how many people attended. It looked from the pictures that I could gather, 15 to 25. Okay, 22 folks attended. The pictures look great. Check out the pictures on the website. Take a look at the albums. So a 1 mile walk through oak juniper savanna, numerous grasses and various flowering plants were observed. Thank you, Ann Adams and Cindy Chrysler, for photo documenting. That's always a great thing to have. And the fall symposium happened just this past weekend, and to give a little brief review and stoke the enthusiasm for upcoming symposia. It's not a very exciting slide, but we have Aaron Buell to get us excited about the symposium.
Thank you. Oh, Jesus, I forgot we have to do that. Okay, we wanted to just let everybody know how the symposium went and encourage people to come next time. It was a lot of fun. It started on Friday with a meet and greet at a brew pub with pizza provided. Saturday, we had choices of maybe 15 different presentations. It was kind of intense. Sometimes there was just one speaker, sometimes there was 4 choices and you could go to whichever one you wanted. There was a silent auction, there was a banquet, there were awards. And then on Sunday we had choices of field trips, and our chapter helped out the hosting Tonkawa chapter by doing a field trip at Berry Springs. So that was pretty fun. Next year, yeah, Gary.
How did it feel being in a middle school?
Oh yeah, it was kind of different than usual. Usually they have it at a hotel that has like a conference facilities, but this time we were at a middle school, and I was kind of doubtful about how that would go, but I thought it actually ended up being pretty perfect. It was nice because there's so many classrooms in different places. The cafeteria was there, so they had the lunch like on a buffet line kind of. It worked out really great. Yeah, it was a really nice school. Yeah. Yeah, now, in the spring, we have a symposium every year at the Wildflower Center, and that one's more of a mini symposium, just one day. Next year, the symposium is tentatively going to be in the Valley, And they have some exciting developments there where the chapter there, I don't know the whole story, but somehow they have a building with a $1 lease. So we'll be the first chapter that has sort of like a visitor center or some kind of a place where people can, I mean, I don't know how they're going to staff it or how that all works, but, and it's right in South Padre. So that would be pretty exciting. Does anybody have any other questions about the symposium? Oh yeah, there were we think 11 of us there if we counted correctly. Yeah. So maybe we can get more there next time. It's really fun. I felt after I left, it was kind of exhausting on Saturday. There's a lot of stuff to do in one day, but it's really motivating and inspiring to see the things that people are doing around the state. You get to meet new people that you haven't had a chance because they're not in Williamson County. So it was really nice. Okay, thank you.
Thank you so much.
Mhm.
Oh yeah, almost forgot. So in a mere 48 hours, 36 hours, I can't do math anymore. I teach AP Physics, but oh well. The in-person plant sale, 11 to 2 p.m. at Southwest Williamson County Regional Park. Tonight's in-person book winner, one of the book winners, we actually have two books. If you attend the sale, I will be able to bring that book to you and hand deliver it to you. It is currently at home on my dining room table. So do attend the sale. Lots of good plants. The latest plant list was updated and posted today. So please come, please buy plants, plant those native plants for our native pollinators and birds. Follow signs to the Corey splash pad and it will take you there. And you know what? What's better than attending the plant sale? Well, uh, hey, yes, volunteers. So there we go. We still need volunteers. See Nancy Pumphrey. You can send an email on the website and we'll direct your information to Nancy Pumphrey. But please, it's a fun time. The Round Rock High School kids will be there helping out. They've got good, if squirrely energy. So it's a fun time, neat people. You will learn your plants and lots of good knowledge to be shared there at the plant sale. Yes, Nancy Pumphrey is the sign-up sheet. So very good. And as always, the Williamson County Landfill Pollinator Garden, third Sunday of the month. No need to sign up. Show up, plant stuff, weed stuff, trim stuff, mulch stuff at times. A bit of everything, and good folks out there to meet and do good work with. Aha, here's the book. This is the one that's on my dining room table, kitchen table. And here's teeming with microbes. This is a signed copy, so we'll have two winners this evening, and it's going to be great. It works out even better when I mess things up. So stay in touch, nips.wilco.org. Sign up for our blog, get those notifications of last minute changes to our location. Chapter meetings, always note the location of the meeting. Last time I said, hey, the next few months we're at Georgetown Public Library, and then we're here now. So always keep note, check the blog posts, check the notifications, but should be at Georgetown Public Library next time. Okay, I'm going to introduce our speaker now. Okay, so Laurie Dixon is a landscape design consultant with a strong focus on ecological principles and the founder of Biotic Lens, a consultancy dedicated to soil microscopy education and applications. She combines her background in landscape design and specialized soil microscopy to help growers and landscapers implement a biological approach. She is a longtime master gardener and serves on the board of the Texas Organic Farmers and Gardeners Association. So welcome, Laurie Dixon. Thank you.
Thanks everyone for coming. I'm so happy to be here. I was telling them earlier, I get to be with my people tonight. I usually speak to farmers and gardeners and growers, but native plants is my first love. That's what I started with. So it's really it's fun to be here tonight. Let's start with a quote. Essentially, all life depends upon the soil. There can be no life without soil and no soil without life. They have evolved together. I wanted this quote tonight because I want to begin with life. Maybe you guys have heard, what's the difference between dirt and soil? Life. Life. Guess who said this? Dr. Charles Kellogg. He was the principal soil scientist of the predecessor of the NRCS, which is the National Resource in our National Resource Conservation Service. He wrote a book in the 1940s. So people have known this all along, right? That the soil begins with life. I mean, life begins with the soil. Okay, but we're kind of having a revival these days, right? People are starting to recover that awareness of the life that we walk on every day. So this is the title, The Soil Food Web Through the Lens of a Microscope. I want to know how many of y'all have heard of the soil food web? Can you raise your hands? This is so refreshing. I give this talk all the time. I mean, out of 100 people, 4. All right, that's wonderful. Okay, how many can name the key organisms of the soil food web? No, there's just 4. No? All right, well, have a look at this photo here. I mean, you guys, I can't move this very well. I'm very interactive, so it's going to take me a while to get my bearings with this equipment. What are those strands? Oh, well, we'll just start here. We have plenty of microscopic images to look at. So this, and it looks like it's not working. There it goes. So why do we care about the soil? Well, I mean, we're all gardeners here because we care about our plants, right? We specifically care about the native plants, which means we specifically care about the native plant communities, okay? That means we also care about the insects, the birds. These are all from my garden, by the way. I did a search on my phone, pull up the wildlife in my pictures, and it pulled up our cat Mo. And I just kind of want to say something about this plant here. Y'all recognize it, right?
Yeah.
Kidney wood. That's a keystone species here. If you do all your research on keystone species, it's hard to find the ones for Texas because everyone forgets about us, right? So we kind of got to do our own thing. Kidney wood. This is covered in all kinds of pollinators, and you see that lizard, the green anole. I know some people like to say anole. I say anole. So if we want healthy soil, we want a healthy plant. What comes first? The soil or the plant? It's a cycle, right? I always keep thinking I'm going to rework this slide to be an upward spiral because that's what it is. Life begets life, right? If you have healthy soil, healthy plants, we'll also have healthy people and healthy planet, right? It's all related. I wanted to start with this. We're going to start with plants because we're talking about the soil food web, right? But do these microbes just live in the soil? Nope, they're everywhere. They're in the air. They're in the phyllosphere, which is the leaf. They're on the leaf, in the leaf. There's an endophytic microbiome. Those are the endophytes that are actually in the stems, in the roots. We have the rhizosphere microbiome. That's rhizo, meaning root. Sphere is the area around the root system. And then the soil microbiome. Okay, we'll be looking at images of microbes in most of these zones except for the air. Maybe not the phyllosphere. I don't think I have anything. This is what I get excited about. You see let's see, rhizosphere, the blue lines going down to that area around the roots. That's where a lot of exciting research has been coming out for the last, I would say 20 years, but really it ramped up in the last, 10 years. A lot of research. So we will be talking about that rhizosphere. These are plant-dependent microbes. I mean, we can't, we're going to talk about microbes tonight. We're going to see a lot of images, but we can't forget that this is an ecosystem. It's an ecosystem beneath the ground that's connected to the ecosystem above the ground, that's connected to the greater ecosystem of the whole earth. So what is the soil food web? This diagram, I consider it kind of old school, but I like to give credit where credit is due. A lot of the research behind this diagram was being done by Dr. Elaine Ingham in the 80s. And it got popular when the USDA published I believe it's called the Soil Biology Primer. Soil biology, yeah, I think it's the Soil Biology Primer, and they published that in 2000. And this diagram got popular at that time. We're going to revisit this again, but I want to kind of zoom in on what's going on here. You can see we have the sun shining, we have the plants doing their thing, we have all the different microbes below ground, and then you can see the players above ground, the higher predators. But let's start with photosynthesis. I hope no one rolls their eyes. Maybe you think you know photosynthesis, okay? But, let's go through it here. The plants are taking solar energy, carbon dioxide from above ground. Those are the only two nutrients that aren't below ground. I mean, there is CO2 in the ground, but the plants don't draw from that. So they're taking those two elements, and then they're grabbing water from below ground, and they're using that. They're turning that solar energy into chemical energy, which is really just carbohydrates. That's a very, very simplistic, it's a lot more complicated as what they're producing. But they are producing these carbohydrates, which are simple sugars and complex sugars, just like, us. And what they're doing is they're sending up to 40%, and some estimates are even higher. They're sending that energy, let's go back, down through the roots, out the tips as exudates, exudates. And what does it say there? It exudes these carbohydrates to what? To feed the organisms. So, really the plants are in control. They're the ones able to do this, to capture that solar energy and convert it into food for us, food for the microbes, food for the whole world. Dr. Christine, why can I not remember her name right now? Dr. Christine Jones coined the term liquid carbon pathway to refer to this process. And it's carbon up in the sky getting converted and going down into the soil. Why would plants do that? they're not using it to grow, they're using it to feed all these guys down here. Carbon is the currency of this economy. The plant is trading that carbon for nutrients in the soil. The microbes are going out in the soil and mining the nutrients. Sometimes we say the plant is farming the microbes, and the microbes are mining the soil to bring back to the plant. We're going to go even deeper. That's not all, folks. There's more. So now let's look. This is a root. A root. Yeah, I missed my... So that's the root tip down here. We call it the meristem tip. And the microbes in the soil, this is another nutrient pathway for plants to get their nutrients. The microbes are down here. I did it again. I don't know what I'm doing. The microbes are down here. They're going in through the root tip. Some of them burst, and those are nutrients for the plant. Some of them don't burst, and the plant does its thing, breaks down the cell wall of these bacteria. They don't have a cell wall, but then it expels them through the root hairs. Is it on?
Try that clicker.
Oh, it's a little tiny thing, but can y'all see it okay? Yeah. All right. So, for a long time, we thought the root hairs were the extension of the plant, right? And it was how the plant was reaching out farther through the root hairs. Not so. Those root hairs, the growth of the root hairs is initiated by this process. Once it strips the cell walls off the bacteria, it releases another hormone which initiates the growth of these root hairs, and they exit through the root hairs back into the soil. That's the rhizophagy cycle. The ones that discovered it, rhizo is root, phagy, eat. The plant is eating. I mean, there's actually, for real, there's a discussion these days that plants aren't vegetarians. So I want to look here. So here's a microscopic image. That's the root tip. And these are the exudates leaking out of that root tip. And Ray Archuleta calls that liquid sun. But I'm going to be throwing out a lot of names. So if y'all want more information, write down those names. You can find them all over YouTube. And this next picture, I love it. This is in the book that you have on your coffee table. I believe this is a confocal microscope. It might be confocal electron microscope. This is the root tip. And these are the exudates leaking out into the medium here. And I love this picture because it reminds me that light is being converted into life. And where is that? That's in the soil, where there's presumably darkness, right? But no, the plants are bringing that light into the soil. Okay, so I don't have the fancy confocal microscope, but I think I have a pretty cool setup. This is, at home. This is on the road. I am certified through the Soil Food Web School as a lab technician to do my own biological soil assessments. It's just a standard compound microscope in every biology lab in the U.S. in high school, whatever. Okay, but yeah, Nancy, she says standard. The microscope is standard, but yes, it does have a lot of bells and whistles. And here's the equipment. I have a camera, a 4K camera, and so you're going to be seeing a lot of those images tonight. I have an epifluorescent module, that's right here. That allows us to see in a certain light range. It's cyan blue. And when you look at samples under the microscope and you flip that on, phosphorus will fluoresce green. And a lot of, especially the fungi, that's what they do, is process that phosphorus. So some of these organisms will fluoresce. And we'll see some of those images. And then I also have this dark field condenser, which is down at the bottom. And that allows us to look at a sample and see it with a more natural lighting. And it's an oblique light. It's just the light coming in from the side. And yeah, so far this is it. So this is with the dark field condenser. This is what is this? It's part of the sunflower. It's a piece of sunflower, the petiole of a sunflower, and I sliced it. Didn't do anything else. Put it in a well slide under the microscope, and use this natural lighting. And this is everything that we can see. And it potentially means nothing to y'all right now, but you can see these are in the plant tissue. These things that we're seeing, the sunflower was starting to decline, so you can see those the senescence in there, that gradual dying. Let's see if it'll play the next one. All right, this is the same sample, the exact same thing under the epifluorescent module. And see now, look at that. Look at what you can see now. Those are probably mold spores, the little round with spikes all around them, which makes sense. The sunflower was at the end of its life. It was one of the big native ones. And we can see, again, I know this doesn't mean a whole lot right now, but you can see what's going on inside of it. So it's going blurry because I'm moving, I'm going in and out of depth. When you have a slide on the deck there, you have 2 pieces of glass, right? But you still have 15 layers, okay? So we can see the top and then go further down, come back up. And there's a lot of techniques to kind of get the lighting right so that these things kind of pop and you can recognize them, what they are. Let's go back to the soil food web diagram. We're going to talk about what the 4 key organisms are. Let's see if I can get this. There's only 4 that we're going to focus on. Bacteria. Here's the bacteria here. They are, if y'all remember trophic levels from food chains, they're the second trophic level. So the soil food web really is like a food chain, except more complex, right? It's not linear, it's a web. And all the different connections are who eats who, right? In a chain, it's who, this one's eating that one's, well, in a web, you'll see. The bacteria are the first trophic level. There are some photosynthetic bacteria that would be in this first trophic level, but with the soil food web, we're mostly concerned with these, that second trophic level, where they're getting exudates, they're getting nutrients from the soil, from the exudates, and they're storing them in their body. They're not releasing them, they're holding them. Next is fungi, also second trophic level. Again, the fungi are decomposing, they're holding those nutrients in their body. That will make more sense as we go on. And then we have the protozoa. We've moved up a trophic level. These are the predators. Okay, now we've gotten to the predators. So they're eating these guys, among other things. And then the nematodes, okay? Look at here, the nematodes, they're eating all kinds of stuff. We have root feeders, we have bacterial feeders, fungal feeders, and we have predatory nematodes. They're eating anything they can fit in their mouth. So what are we looking, what are microscopists looking at when we're looking at these samples? Well, we're looking at those 4 key organisms. We want to see all of them, all of them. And we're looking at aggregates, soil aggregates, because that's very important with the soil, right? The aggregation in the soil, it's basically the difference between having a pile of sand or a sponge, right? And you can imagine, I mean, you imagine a pile of sand here and a sponge, and you pour water on both, what's going to happen? The sand is going to shed it, right? But the sponge is going to soak it all up. Okay, so aggregation is significant. I mean, we have to have aggregation. So we'll see some of those in the slides. Occasionally we see something interesting with the background color. We're looking for diversity. Not only do we want to see all of them, we want to see different kinds of them. And then we're also looking for these aerobic and anaerobic indicators. And here's some other stuff that we see. So let's start with bacteria. When it comes to bacteria, obviously we can't identify them to species with a compound microscope. You would need a DNA analysis to identify them. But what we can see under the microscope is we can see their shapes. That's their morphology. Morphology is important in the work that we do with all the microbes. So with the bacteria, you see little circles, rods, these funny shapes. You see the circles chain up, the rods chain up. And I chose these 2 pictures for a particular reason. These are from a colleague of mine, and she has the minimum requirements needed for a microscope to do this kind of work. And you can see the quality there. For my old eyes, no, I can't do this. But this is what she has. But look, it's good enough. You can see that shape. Doesn't that look remarkably like this right here? So there's a spirochete. Okay, and here you can see little circles. Everything looks white right now when we do a certain maneuver with the microscope. We call it shadowing. These will turn black. But you see the little dots, you see the rods. This is diversity. When I say, what do we see when we look at samples? My samples are just not soil samples. They're also all different kinds of compost, DIY inoculants, commercial inoculants. This is a DIY inoculant called Jadam, and it's used in Korean natural farming. And I had someone he wanted me to look at theirs and look at all of that. This is all different kinds of bacteria, these little guys, there's some that look like they're kind of curving, and it's like, ooh, do we have spirilla in there? But if you look closely, you can see the joints. You can see, no, they're chained up cocci. And you can tell by their movement, it's kind of stiff, right? But let's see. So the other thing, remember, that we're looking at is it anaerobic or aerobic? Why do we care about that? We need oxygen. These microbes need oxygen. Plants need oxygen. Most living things thrive on oxygen. We do have a lot of microbes that thrive in anaerobic conditions. Actually, yeasts go all the way. They can be in any category. But aerobic is above 6 parts per million. And these are the microbes that thrive in an aerobic zone. We won't talk a lot about facultative. If you're familiar with Korean natural farming, effective microbes. They use a lot of facultative preps. And these are the guys that live in that zone. And then in the anaerobic zone, of course, you have anaerobic bacteria. You also root feeders. So when we're looking at a sample, these are clues, right? We can kind of see, how is your system going? Is it going aerobic or anaerobic? Which do you think we want? Aerobic, predominantly aerobic. Dr. Tom Dykstra, his research, he's based out of Florida, is that we need about 80% aerobic bacteria of the soil food web in the soil. So this is a commercial microbial inoculant, and I wanted you to see this because what is all of that? All kinds, cocci, rods, that's just bacteria. And I started at the top of the slide. Now we're getting onto the main plane of the slide. You just saw layers and layers and layers of bacteria. This is what we would call a bacterial dominant sample. And that hopefully that will make more sense by the end of the lecture. So with the bacteria, we also have actinobacteria, and that is a filamentous bacteria. So it's a filament. It almost looks like fungi, but it's a lot smaller. I can't tell if it's playing. I don't know if y'all saw the little circles when it'll come back up and you'll see the bacteria in that strand. It's just a single strand. And that's how we identify. And this is at 600x. Most of the images that we've been looking at so far are at 400. So that's quite a bit more magnified. It actually looks like a strand of hair. But actinobacteria are important. They are major decomposers. And so you will often see actinobacteria in the compost, in the soil. They do thrive in higher alkaline conditions. So there are some, they do send us some warning signs if we see too much of it. So let's go on to the fungi. That's the next group, okay? There's a lot that we could say about fungi, and I just want y'all to know, I'm not giving you a lot of details about these organisms, but what you can do if you're interested is go on the internet, look up the organisms, find out what they eat. That's important for anyone who's growing, because if you know what they're eating, you know what their nutrient, you can kind of deduce the nutrient pathways in your system. How are your plants getting their nutrients? So when it comes to fungi, I just think looking at them as far as their functional group is helpful. We have the saprophytes, which are the decomposers, pathogens. We probably know the pathogens more than anything, but listen, of all fungi, what % do you think is pathogenic? How did you know that? 1% are pathogenic. Look, all the rest are beneficial. Okay, mutualists. These are the obligate symbionts. We'll talk more about that later. And the most famous one is the arbuscular mycorrhizal fungi, short AMF. But we'll start with yeast. Yeast is a fungi. And this is a product called EM, and it's a facultative consortium of bacteria, or a consortium of facultative bacteria. It was developed by Dr. Higa in Japan, and it's sold here in the US. But look at, that's yeast. They're chaining up, they're budding. These little rods are probably actinobacteria. Here's a saprophyte, classic saprophyte. It's that long brown strand going across the whole field of view. In this photo, we also have a potentially pathogenic fungi, oomycetes. And you know, oomycetes can, I mean, those cause disease, but so what I would be looking at is, what are the clues here? I mean, is this system going in this direction or this direction? And there are a few more clues in here. It's these little guys. You can't see them, but that's testate amoeba. Remember, those are the predators. And we have good aggregation. These are little micro aggregates. They have a dark color, which indicates to us that it's humic acids, all very, very beneficial stuff. So yeah, there's a little warning here. This is a root. Maybe this plant is in trouble, but overall, this system is looking decent. This is Comanche, Texas, and they have 87% sand. Aspergillus. So this is interesting. It's a saprophyte, but also pathogenic, can be pathogenic. And that's the thing with a lot of these organisms. They can change how they behave in your system depending on the conditions of that system. And Aspergillus is commonly found in compost piles. If you have microplastics, they might show up. They eat microplastics. So okay. So any guesses? Any guesses what this is? What do you think? Organic material? More fungi? Some kind of weird microarthropod? This is where my epifluorescent module comes in handy. When you turn that on and you see in that light range, microplastics will show up red or orange or a really bright yellow. It looks very unnatural, even under the microscope. But that's a microplastic. This was someone's DIY product that they made, a microbial inoculant, and they got microplastics in it. All right, look at that beautiful photo. This is arbuscular mycorrhizal fungi, obligate symbionts. They have to have a host. Again, remember, these are plant-dependent microbes. You take away the plant, the microbes die. They have no food source. They cannibalize one another, or they go away. They have no more food, okay? So the AMF in particular is very important in our soil systems. This is a photo by Sarah Wright, who discovered this stuff. These are spores, okay? This is the root, these are spores, and look at that webby mess all around here. That's glomalin. Gay Brown says glomalin. But glomalin is a substance that AMF produces. It's a sticky protein. It is extremely important because that sticky stuff is what holds the soil particles together. And if you think about the soil and you know, in classic soil science, we have 50% pore space, right? 25% water, 25% air. In our systems, especially in Texas, that pore space is greatly compressed. We have about 1 to 2% organic matter in our soils, vegetables like 5%. Just think about if you don't have that organic matter, you don't have that sponge. What you have is compaction. You have concrete. How many of y'all have dug in concrete? Well, this glomalin resolves that. If you can get AMF in your system, then you can build that aggregation. here's a diagram. And the main thing I want to point out in this diagram is here's some plant tissue. The AMF, their obligate symbionts, their host, they get inside the plant and they follow, you know, these pathways. But look what's happening here. It's a root hair. So the extensions of the root hairs in the soil is AMF. And AMF associates with something like 80 to 90% of all plants, and there's different kinds. This is my image of AMF. Again, I don't have that fancy, you know, but look, this is in my experiment plot. This is the spore. This is the hyphae that's coming, you know, usually this is what's attached to the plant. This is not plant though. This was actually in a wood chip pile. And typically, I mean, you can't grow AMF without a plant. But I think that the wood pile was from oak. This is probably the kind of AMF that associates specifically with oak trees. And it's sporulated in my little experiment compost pile. It comes from that guy. I think it's called it's like a puffball, an earth. It's one of the ones that when you pop it, you see the black, you know, go. Scleroderma is one, but I can't remember the common name that they call this particular mushroom, an earth ball maybe. All right, so we're to the next group, protozoa. We mainly see these guys, flagellates, amoeba, ciliates. There's one thing I really want you to get with protozoa. No protozoa, no nutrient cycling. And just kind of quickly kind of think your way through that. Why? Why is that? Well, these are the predators, right? And the soil food web is really a predator-prey system, right? So those bacteria and fungi are holding all those nutrients. They mind all these nutrients. They're holding them in their body. They don't excrete them. They have extra nitrogen. Bacteria, some of the bacteria eats lots of nitrogen. They're 5-to-1 nitrogen to carbon ratio. But they don't give it up. They just hold it. They got to get eaten. And then the protozoa eat them. And then what do the protozoa do? They don't need all that stuff. They poop it out. We call it the poop loop. Sorry. Okay, this is a flagellate. I named him Mr. Hermit Crab after, you know, the Eric Carle book. You know, A Home for a Hermit, I think it's called. Look at that. So do you see his flagellum there? Can y'all see it? That's really hard to get. You have to get the lighting just right to see that flagellum coming out. It's kind of, they use it for locomotion, right? He's reaching out there and it's kind of propelling himself. Oh, I kind of want to see Mr. Hermit Crab again. Anyway, I'll tell you about Mr. Hermit Crab. You saw what he was carrying around, right? He had all this, it was a little piece of sand. It was kind of reflective, so that was a sandy soil. So he's just going around doing his thing, looking for bacteria and catching on to these little pieces of things. Bacteria is hitching a ride on him. They're like taxi cabs. Another thing they can do in their, you know, in the soil. Oh, 10 minutes. Okay. So now we are to amoeba. Again, these are the protozoa, and you can see these are testate. They have a shell. They make a shell out of mineral material. I want you to see the next one though. This is the one you may have seen in biology class. You know, no shell, right? Kind of amorphous. Isn't he beautiful? So no shell. The other ones have a shell as a kind of protective mechanism. Ciliates. The cilia, they're named after this cilia there, little faint hairs, just like the flagellum of the flagellates. They use that for locomotion. They also have a kind of sensation with these hairs. Ciliate in action. Again, a really hard video to catch because these guys are fast. He is doing something we call bacterial grazing. He's just going all around those aggregates. You know, the bacteria hang around those aggregates, and he's just yum 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 yum. Nematodes. We know this guy, the root feeders. Again, very, very small percentage. Most of the nematodes are beneficial. We want them in our system. We need them in our system, okay? So we look at nematodes, again, with all the microorganisms, we're so concerned with what are they eating. So these are all named by what do they eat? Roots, bacteria, fungal. He has a little spear there to pierce the strands, and then a predatory, and he has that big giant mouth. He just eats whatever he can stuff in there. I just wanted to show this. These are the microarthropods. I mean, they just look so crazy. They are small scale grazers, big impact decomposers. They do a lot of work in the soil. This video is a rotifer. So fun to watch. There he is. He has a little kind of fast hold that he attached to this aggregate. Is holding him stable. And look at him. He is just gobbling up everything he can find around those aggregates. This is a testate amoeba. He's just like, ooh, getting bumbled around, you know. I don't know why he's just there. But this guy is eating, eating, eating. Oh my gosh. That's a mite. Yeah. A microarthropod. This was in a compost. So y'all got any questions? Yeah, go ahead.
You mentioned that there are certain organisms that when they enter the plant that their membranes get broken down, but yet then you talked about, like, I think the fungi where they stayed in. So are plants selective in what they break down?
That's a good question. I'm not sure. They are selective. And that's why I say plants are in control, because they can change the composition of that exudate based on what they need. Those exudates, plants are communicating with microbes all the time. And we know some of the ways that they communicate. We definitely know they communicate, but we don't know all the ways. But exudates, there's chemical signaling. They are sending out the message. I got this, guys. I got hamburgers. If you read this book, that's how Jeff always likes to say it. I got Chinese food. I got hamburgers. Who wants the Chinese food? And so, yeah, they are being selective. But as far as the two that go in, basically what I was referring to is two. Some of the bacteria, when it takes them in, some of them just kind of, I don't know what happens. I mean, maybe like osmosis or something, when I don't know, when things explode, but they just don't make it. They don't make it, and they burst within the root, and those nutrients are released. But others do make it. You know, and I don't know. I don't know what's the difference. Why do some make it and they don't, and some don't? But we know that that happens. Yeah.
You mentioned something about Korean farming?
Korean natural farming.
And tell me about that.
So Korean natural farming, I'm not an expert. I just look at the samples. But it was, Dr. Cho. Was it Dr. Cho? Yes. Developed that. And they use what they call effective indigenous microorganisms. So basically they have different preps. The first one is you do a prep, a certain kind of procedure to go collect microbes in a healthy system. So the biggest, best oak tree, and you put parboiled rice in a box, etc., bury it in the ground, all different kinds of things. You collect the organisms, you bring them in, and then you do another, you cultivate that, you do another prep by adding foods and doing, and they use those preps for a lot of different things, for nutrients, for biocontrol. Yeah. It's very interesting. Yeah. Any other questions? I do have, if we have time. Nope, no time? Okay. No questions? Okay. Yeah.
Because I was curious about this, because you saw on the bottom of your slides that they were inoculants. So when they're inoculating the soil, like how much do they need and what do they do? Because I imagine like for natural farming using this method, it's not like you're just
Right.
putting what you how do you like how much and how do you get this in? Is it a water addition that gets put in irrigation or?
Right. So with Korean, she's asking about how do you gauge the amount of inoculants that you're using. With Korean natural farming, I mean, they may or may not be using the microscope. They have a lot of observational indicators that they're using, and I can't really walk you through that. But for us, we look at the soil under the microscope. We count the organisms, and we calculate a fungal to bacterial ratio. We know how many predators. Of course, they're all estimations, but we know what your system is missing. There are all kinds of ways to add that biology back into your system. And with the Jadam was one of them. They're kind of like throw everything in, including the kitchen sink. And that's why users just look so crazy. That philosophy is we want as much diversity as we can get, and we're going to let nature sort it out. And, obviously you want a healthy system. You don't want to spread disease or whatever. But again, remember, the percentages of the pathogenic ones is really small. Yeah. And if you regulate, how much food is in the, whatever it is, the inoculant that you're using, and how much air is in there, aerobic versus anaerobic, then you can kind of control those things. Yeah. Any other questions?
So that means you gotta have different inoculants for different place soil?
Oh, that is such a good question.
Can you repeat for us?
So that means you need to have a different inoculant for different soils. Kind of, yes. And you also could potentially need different inoculants for different plants, because those mycorrhizal, the fungi, there are different species that associate with different plants. And when it comes to native plants, I mean, the research isn't as big. No one's looking at these plant microbe associations with native plants. I don't want to say no one. People are most research is centered on agricultural crops, right? As they should be. But native plants, we also need that, right? I mean, I'm an ecological landscape designer. We need to understand this, and we need to understand how we can have healthier native plant communities and get them established more quickly. So yeah, there's a lot behind that. I cannot give you a short answer. That is worthy. I give workshops that are, the minimum is 4 hours. 4 hours minimum. And we never get to all that. I mean, yeah. Any more questions? Well, how much time do we have?
Not much.
Okay. Well, I'm just going to introduce this word to y'all because this is going to connect what's happening below ground with what's above ground. And it's succession. Did you see the sequence of changes in an ecosystem after disturbance? So at the beginning of the earth, nothing, okay? Then plants went through succession. When there's fire, when there's drought, when we, raise it with a bulldozer, that's disturbance. And these microbial communities, this will speak to your question. They change as the system goes through succession. At the beginning, you have dirt, bare soil. You have a very low fungal to bacterial biomass. That means you have lots of bacteria, okay? And then it goes up to weeds, early successional, mid-successional. A lot of our wildflowers are early successional, but some of them are even these pioneer and ruderal species. And these things below ground that are changing as the plant communities are changing above ground. Ruderal is just the word for the guys that come in at the very beginning. Yeah. And the characteristics of a bacterial dominant soil are going to be pore structure. This is it. This is it. If you have lots of weeds and cracking in your soil, you have a bacterial dominant soil. This is what I see everywhere, day in and day out, okay? Higher successional plants aren't going to do well in this because they prefer a different form of nitrogen and they need more fungi in their system for a lot of reasons. This is a bacterial dominant soil under the microscope, very crowded, lots of bacteria. This is what it looks like above ground, okay? I'm talking about this here, just dirt, they mowed it, not covered. This is bacterial dominant, and you can tell by these plant communities that are here too. And then you go up in succession, to productive pastures, to shrubs. So don't think that this doesn't matter to native plant people. I mean, we want our shrubs, right? This is a low successional garden. These are low, not low, early successional, early. Was I saying low? Early. These are common in early successional communities. This is my winter cover crop. This is what I have covering my ground through the winter. Cilantro, parsley, cordalis. This is a transitional plant community. We have some annuals, some early successional, but then now we're getting into the clump grasses, the little blue stem. This is a transitioning native pasture. He's, no chemicals, laid off all the chemicals, doing a lot of things, and it's showing. Look, what do you think you want? What is this? Mid-successional. Shrubs, vines. This wants a more fungal dominant soil. I can stop whenever you say stop. If you're more interested in soil biology with design, ecological design, these are all Bibles to me. These are not just recommendations. I use these all the time. This is the one I've used the longest. This is where I came, and when I came back to Texas after a short, regrettable stint in California, this is the book that I got. Native Texas Plants by the Wasowskis. Both passed away now. But these are all design books. If you're more interested in the soil, these are the soil books. Again, I have to admit I haven't read this entire series, but I do know that most of what I've talked about is in those books. This is my Bible and these. If y'all have any questions, you want to contact me?